Today we will discuss allosteric enzymes. Allo means other, and as these kind of enzymes have one other site which is not similar to the active site or the substrate binding site of enzyme, these enzymes are called allosteric enzyme. Now these enzymes are also named regulatory enzymes because the activator or the inhibitor serves as regulator which can either upregulate or downregulate one reaction. Now binding of the effector induces conformational change in the enzyme which helps other effector molecules to bind to the same site for increasing its catalytic efficiency. Now these kind of behavior is also called cooperative binding. Now the allosteric enzymes utilizes binding energy to carry out this conformational changes. We have seen in our previous video that the enzymes normally uses the binding energy to lower the activation energy, but in this case most of the binding energy is utilized in changing the conformation of the enzyme for carrying the reaction out. Now ATPase or aspartate transcarbamylase is one enzyme which is a classic example of allosteric enzyme. Now CTP serves as its inhibitor and ATP serves as its effector. Now when the effector binds to the allosteric site, allosteric activation occurs, but when inhibitor binds, allosteric inhibition occurs. Now these kind of enzymes are very useful in explaining the feedback regulations, which we will discuss later on. This is the kinetics of the allosteric enzyme. This equation is known as the Michaelis-Menten equation. We have discussed it in our previous video, and this is the Hill's equation. Now what happens is, the difference between these two equations is this n factor. So this n denotes the number of site. Now the value of n ranges from 1 to 4, typically ranges from 1 to 4, and the conformational change which occurs in the allosteric enzyme due to the binding of the effector and the inhibitor changes the hyperbolic nature of the Michaelis-Menten curve to to sigmoidal curve. This is the basic difference between these two. Now coming to the feedback inhibition. And what is feedback inhibition? So this is enzyme 1 which catalyzes this reaction, enzyme 2 which catalyzes this reaction, enzyme 3 which catalyzes this reaction. Suppose this is A to B, B to C and C to D, which is the final product. So in case of feedback inhibition, what happens is the product or the D inhibits the first enzyme of the reaction, which is enzyme 1. And what happens in case of feed forward activation? As from the name itself, that this will activate the enzymes which are participating in the reaction down the line. So in case of feed forward allosteric activation, an intermediate of this pathway acts as allosteric activator of the enzyme catalyzing the later steps of the reaction. So suppose this C is the intermediate which will act as allosteric effector for these enzymes to carry out their reaction in efficient manner. So this is all about the allosteric enzymes. Hope this will be useful for the students. Thank you.